Hello everyone, welcome to Learn Bioinformatics from Scratch. This is part 5, and today we will learn how we can deal sequence data and how we can perform different kinds of analysis. My name is Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. Part first, we learn about sequences and sequence retrieval. In part second, we learned about alignment and the alignment algorithms and types. In part third, we learn about homology and different types based on homology. In part fourth, we learn how to use BLAST and the data we downloaded during part 4 will be explored here with different programs. So we will use two programs, both are freely available online. One is BioEdit and another one is Mega. We will explore both these programs one by one and we'll see what we can achieve data or what kind of analysis we can perform from BioEdit and Mega. So let's start. First, you have to open Google and type BioEdit software or BioEdit download, whatever. You will see this one, BioEdit download, BioEdit download. Just click here. And once you see, don't click this one because this is a kind of advertisement. And come here, download free 12.4 MB. So it's a smaller software. It will be downloaded to your system, the download folder. Then from the double click, you can install it into your system. Again, how you will download Mega? You can just type Mega 7 or whatever the current is, I think Mega 11 or Mega 12. Okay, Mega 12 beta. So click here download. It will be downloaded and then you can install with the double click and use this program. So I have both the programs installed already. So therefore, I'm not going to reinstall it. It will take almost until 5 minutes of you to download and install. So now let's explore these programs, how they will work on our data. I suppose or I assume that you have watched part 4. So this is the file we downloaded in part 4 and it contains all the sequences downloaded from BLAST. So let me copy it to desktop. By the way, it is not necessary, but it's okay. Okay, then type here BioEdit. It is installed in my computer. Once you open the software, you will see this kind of black window. Just click here, File, Open, and then click Desktop because my file is located here. NDM1 blast.txt and open. Based on the sequence quantity, it will take that much time. Now, these are unaligned or raw sequences we downloaded from BLAST. Now, first of all, let's briefly explore this BioEdit, then we will learn what it can perform. Okay, there are total number of 94 sequences we uploaded or our dataset contains if you drag this part, this is the naming panel, and the name of each and every sequence starts from the accession number, then the Klebsiella pneumoniae strain, NDM1 gene, blah blah. So almost each and every sequence contains a long stretch of name. So if you are going, or if you will use, or if you intend to generate a phylogenetic tree, then this kind of naming system looks awkward. So you will need to to edit or to minimize these names. How will you do? So you can also do it in the text file, or you can also perform it manually, just like if I'm clicking here. So you can see it opens only this sequence. So you can just, if I want to remember, if I want to delete each and everything, just keep, let's say, K pneumoniae or Klebsiella pneumoniae, K underscore pneumoniae, or also I will connect the accession number with underscore. So just apply and close. Now see, this name is changed to the shorter version. Secondly, you can also open this text file and change it here. Let's say I'm removing this. Let's say I have removed two things. Okay. One thing I mentioned in part first, do not remove this part. If you remove this part, this is the header sign, the program will be unable to read your sequence. Okay. So, and you are allowed to, if you just want to enter only K, let's say K, I think I misspelled it. Okay, K dot P. So it's okay. The program will read it. Now save it, and I will re-upload this data to show you how it will work. Okay, file open, and now NDM1, you can see those 94 sequences. The first one is KP, and the second one only contain the accession number because I removed, I only fixed these two. So overall, if you are planning to generate some pictures or to generate the phylogenetic tree later on, then it's better to handle the sequence naming at this stage. I taught you how can you handle within the BioEdit, or how can you handle in the uh, text file. Okay. If you see these color bars, it tells you, it changes the color. Let's say if you click here, you can see. So these are the different options. If you want to explore, you can. There are some other coloring scheme. Okay. Next thing, the next important thing is to explore these things. If you want to delete some sequence, let's say I have selected these three sequences, and now I realize that these three sequences should not be here. Okay, then how to delete it? Just let's say select here. Okay, now come to edit and say delete sequences. Now you see the total number of sequences are now 86. So I have deleted some sequences just to tell you how can you do it inside the inside this BioEdit. Okay. Also, you can explore this edit tab, and you will see many other options. This is the sequence tab. It also will give you a lot of options, and if I started to teach each and everything, it will make the video so longer. So I'm not going to do it. You have these are easy things to just read by yourself. But the most important thing, I do not want you to skip it, is the accessory applications. So if you want to perform alignment, so you can click here and it will say cluster W multiple alignment, and just then click it, run cluster W. It will align the whole data. I'm not going to align it. Okay, I'm going to align it, but before that I just want to uh, remove some sequences. So let's see. Oh my god, wait, let me load the data again. I just Okay, so I'm going to delete some sequences, and I will click here, and then I will click here with shift, and then I will come here, edit, delete sequences. Okay, now I have total 13 sequence. Why I deleted all these sequences? Because I want the alignment to be slightly quicker. Okay, now click here and cluster W alignment, then click run cluster W. You will see this option. Okay, and once you click OK, it will take some time. Let's see. Yes, it has started. You can see this window. A new window will come, and it will take some time for 13 sequences. But still, if you want to be more quicker, so you can use Mega, we will use after this. Also, you can use some online servers and perform alignment. But why I'm insisting, or why I'm using alignment within the BioEdit, because I want to teach you next is like how to trim your alignment. So I'm going to pause this video for a while until the alignment is finished. Now, once the alignment is finished, you will see this kind of window. This was the this was the real window having 13 sequences. This is the align window. So now you can see the gaps and all those things. Okay, you can close this window and keep the align window open. Okay, now you can see the gaps, also the those regions which are matching, not matching, you can see. Okay, now let's say you want to remove this region, and this is your reference sequence k.p. Let's assume. So how will you remove it? So the right or the right way is to edit sequences after alignment. So now our sequences are already aligned. Now you can remove. Let's say I'm removing this region. So how you will remove? Just first click here. Now click here, but before clicking, press the shift button and click. It will select all the sequences. Now press the delete button on your keyboard, which is above the backspace. So now you can see it's deleted. For example, you are deleting only this option, this place as well. So just click and press delete. Can you see? Okay. So the BioEdit also offers you this kind of functionality, and you can easily handles bigger alignment, smaller alignment, and you can easily perform different kinds of editing. Okay, now file, and then save this file as save as, let me save it, let's say ndm underscore alig, alig for alignment, and it will be saved in download folder. So just say okay, and please check the FASTA option here. It will always be FASTA, but still confirm before saving. So save. Now you can see, let me check the download folder that whether, yeah, you can see ndm alec.fas. Okay, so this was all about the BioEdit. You can also generate some tree file using the BioEdit, but not all the BioEdit tree options are viable or easy to use, some of them. So you can try one by one and you will see it. Okay, now we are going to explore Mega 11. So let's say I am uploading that aligned file. So open and download, and here is the file ndm underscore alec.fast. Click here, open. They will ask you, you want to analyze or align the file. So if you remember, we have already aligned this file. So if you realign this file, it may disturb the file, and you it like it will generate, possibly it will generate more gaps. So that's why you have to click the analyze button, do not align. But if you did not use BioEdit for alignment or any other online software, then in this case you can click align. Okay, now they are asking you, your data is nucleotide sequence or protein sequences. So my data is nucleotide sequences, so I say okay. Then they say it's a protein coding sequence, so yes. Then they say what, like for different organisms, the translation or the codon values are different. So I'm choosing, let's say, the standard one. Now, your data is here. Once you click here, you can see your aligned data. You can see here. And once again, this panel from here, you can perform different tests like different statistic, nucleotide composition, nucleotide frequency, codon usage, and you can highlight different conserved sites, different groups, display, you can change the display stuffs, and you can also close it. Okay, if you click here, it will close the data. Now also the Mega offers different kinds of analysis. So if you are interested in these analysis, just click here, and if you are interested to calculate some pairwise distances, you can calculate here gene diversity and phylogeny. And for the phylogeny, actually we are generating a phylogenetic trees, so you can work on it, and it will you will find it sufficiently good and great, and you can also explore these things. So let me close it. Okay. Okay, so this was all about like how you will handle your data, how you will handle different sequences, editing different sequences. I hope you enjoy this session and you have learned something new. Thank you all for watching these lectures, and I'm waiting for your feedback. Thank you so much. Bye bye.